Bismillahirrahmanirrahim. Assalamu alaikum, dear students. Today's lecture is about the conduction system. At the end of this lecture, you will learn different components of the conduction system, mechanism of self-excitation, and action potential in the sinoatrial node. Also, the regulation of sinoatrial nodal activity and the clinical importance of pacemaker activity of the SA node. The conduction system is a group of specialized muscle cells in the walls of the heart that signal to the heart muscle to contract. It is composed of different components that are number one, the sinoatrial node, then three internodal pathways, 1, 2, and 3, then atrioventricular node, then bundle of His, then bundle branches, that is right bundle branch and left bundle branch, and finally is composed of Purkinje fibers. Conduction system, by generating electrical impulses, controls two things. Number one, it controls heart rate. Number two, it controls force of contraction. So that the blood can be pumped. The conduction system has two very important capabilities. Number one, the self-excitation. It means that the cells of conduction system are capable of spontaneous generation of impulses. Number two, the rapid conduction through the different components of the conduction system. The cells of the conduction system rapidly transmit wave of excitation from one part to the next part through gap junctions present at intercalated discs. First, we will talk about the sinoatrial node. The cells in the sinoatrial node are small, and flattened. These cells are located in the supralateral wall of right atrium. These are located in the supralateral wall of the right atrium, as you can see from this diagram, just superior or just near the opening of the superior vena cava. Two types of cells are present in the sinoatrial node, and these two types of cells are modified cardiac muscle cells. These cells have few striations and indistinct boundaries. The second type of cells that are present in the SA node are P cells, that are called pacemaker cells. These are round cells with few cell organelles and these cells are connected with each other through gap junctions. If we talk about the action potential is, we can say, it is different from other cardiac muscle cells. It is different from other cardiac muscle cells in the sense that it has unstable resting membrane potential, and these cells, due to unstable resting membrane potential, these depolarize spontaneously. As they depolarize spontaneously, they generate impulses. When they generate impulses, that impulse basically travels through the whole heart, and when it travels through the whole heart, each impulse then triggers next impulse, and the next impulse then triggers the next impulse, and in this way this cycle remains in continuation. The reason of this unstable resting membrane potential, that is minus 50 to 60 millivolts in SA-nodal cells, is that these cells are more leaky to sodium ions as compared to other cells. Due to more leakiness to sodium ions, the depolarization in the SA node, it starts slowly and threshold point is reached slowly. And when threshold point is reached, then a rapid depolarization occurs, and this depolarization occurs up to plus 5 to plus 10 millivolts. This rapid depolarization is followed by rapid repolarization, then when rapid repolarization occurs, once again RMP becomes unstable. When it becomes unstable, uh, the membrane again becomes more leaky to sodium ions, then again depolarization, and repolarization occurs, and this process continues throughout life. Ionic basis of action potential in SA node. Before going into discussion of ionic basis of action potential in the SA node, first we will define what is action potential. And action potential is a rapid change in the membrane potential. It is a rapid change in membrane potential by movement of charged atoms, also called ions. We will discuss ionic basis of action potential in the SA node under 5 headings. Number 1, the resting phase. Number 2, pacemaker potential or pre-potential. Number 3, diastolic depolarization, then repolarization, and then cycle repetition. The resting membrane potential in the SA node, as we discussed, is between minus 50 to minus 60 millivolts. And as we mentioned, it is unstable, and it is unstable due to more leakiness of the membrane to sodium ions. When action potential starts in the SA node after resting phase, the resting phase is achieved after repolarization. When membrane repolarizes, basically due to movement of potassium ions, the SA nodal membrane becomes hyperpolarized. So this second phase, that is pre-potential or pacemaker potential, it starts when membrane hyperpolarizes. When membrane hyperpolarizes, then a type of channel opens that is permeable to channel opens that is permeable to both sodium and potassium ions. As the potassium outflux stops or efflux stops, the sodium is more in the extracellular fluid. So the sodium movement through this channel starts. Due to resemblance to hyperpolarization phase of action potential, this channel is called H channel. But due to its funny opening, this channel is named as also F channel. F means funny channel. When this channel opens, the sodium starts moving inside. As you can see from this diagram, when sodium starts moving inside the cells, this completes the first part of diastolic depolarization. When sodium moves inside the membrane, becomes the interior of the membrane, it becomes positive, and when it reaches near threshold value, then voltage gated, then this leads to the opening of voltage gated calcium channels. There are two types of voltage gated calcium channels in the SA node. The first type channels are called T-type calcium channels. The second type of voltage gated channels in the SA nodal membrane are called L-type calcium channel. The T stands for the transient, means they open for a fraction of seconds and then these channels closes. When the T-type calcium channels opens, they complete the second part of the pacemaker potential or pre-potential, as you can see from this diagram. When the pre-potential phase is complete, then the threshold point is reached, and this marks the opening of long-lasting calcium channels that are called L-type calcium channels. Then the calcium influx starts, and this completes the depolarization phase of the action potential. This completes the depolarization phase of the SA nodal action potential. When the depolarization is complete, the L-type calcium channels, they become inactivated. These becomes inactivated. When these becomes inactivated, this causes the opening of potassium channels, and then potassium efflux starts, and due to this the membrane potential again it returns back to resting membrane value, and it even becomes more negative than the resting membrane potential. Means the membrane, the acinodal membrane, it becomes hyperpolarized. It becomes hyperpolarized. This hyperpolarization then again marks the opening of F channels. When these channels opens, the sodium influx starts. When sodium influx starts, this marks the beginning of new action potential in SA node. When this occurs, new action potential, then again membrane repolarizes, and this cycle repeats again, and it remains in repetition throughout the life of a person. This type of action potential that we studied in the previous slides, in this type of action potential, we have seen that the membrane is more leaky to sodium and calcium ions. Now the question arises, if the membrane is more leaky to sodium and calcium ions, then why does this leakiness to sodium and calcium ions not cause the sinus nodal fibers to remain depolarized all the time? The answer to this question is twofold. There are two reasons that why this state of depolarization does not persist. There are two reasons. Number one, the L-type calcium channels become inactivated as they remain open for only 100 to 150 milliseconds. The second reason is that at about the same time, greatly increased number of potassium channels open and large quantity of potassium ions diffuse out of the synodal fibers. This type of resting membrane potential between action potentials is called diastolic depolarization. So, in diastolic depolarization, why it is so called? Because the membrane first hyperpolarizes, then there is slow positivity inside the synodal fibers that reaches the threshold value, and this segment is called diastolic depolarization or prepotential. Second question that comes in our mind is that why is this new state of why is this new state of hyperpolarization not maintained forever? The answer to this question is that during the next few tenth of a second, when action potential is over, progressively more and more potassium channels close. When they close, the state of hyperpolarization also stops. The second reason is that the inward leakiness to sodium and calcium ions again increases, and this increase causes the resting membrane potential to drift upward once more, finally reaching the threshold point, and this is the point for discharge at an action potential of about minus 40 millivolts. Then entire process begins again and again. Self-excitation. This is why we called that the synodal fibers have the property of self-excitation, and this self-excitation then leads to generation of new action potential. Then cycle repeats again. As we have discussed that the synodal activity generates impulses. Synodal activity generates impulses, and these impulses travels to whole myocardium, and contraction of heart occurs. On the basis of this, we can say that the SA node, it controls heart rate. The heart rate depends on the rate at which the SA node produces action potentials. That is, it is between 60 to 100 beats per minute. This activity of the SA node, it results from two set of nerves. It results from two set of nerves. Number one, the sympathetic nerves. Number two, parasympathetic nerves. Sympathetic nerves begins in the thoracic part of spinal cord, specifically between T1 to T4 segments. And these sympathetic nerves, they release norepinephrine neurotransmitter at their nerve endings, while the parasympathetic nerves supplying the SA node, these originate in the brain and pass along vagal nerve fibers. That is the 10th cranial nerve, and along the vagal nerve fibers, at their endings, these nerve fibers, they release acetylcholine as a neurotransmitter. So the impulses from the, or you can say the change in the heart pressure or the SA nodal activity, they passes through 9th cranial nerve and through the 10th cranial nerve to the cardiovascular center located in the medulla oblongata. Then, if the heart rate is less, then the sympathetic discharge increases and it passes from the cardiovascular center to the thoracic segments between T1 to T4. And then from these segments, the impulses pass through the sympathetic nerves to the SA node and also to the atria and also to the ventricles. In this way, it controls both heart rate and force of contraction. While the parasympathetic nerve fibers, they pass along the vagus nerve and through vagus nerve they pass to the SA node, and at their nerve endings they release acetylcholine as a neurotransmitter. First, we will discuss the effect of the sympathetic stimulation on the heart. As we have mentioned that the sympathetic nerves, when they are stimulated, they release norepinephrine at their nerve endings. When the norepinephrine is released at their nerve endings, the norepinephrine then binds with the beta 1 adrenergic receptors present in the SA node. The binding of norepinephrine with the SA node, it causes activation of G stimulatory protein. The G stimulatory protein, after activation, it leads to the production of cyclic AMP as a second messenger, and the cyclic AMP in turn increases the permeability of SA nodal membrane to both sodium and calcium ions. When this permeability increases, it causes a more positive change in the resting membrane potential, and it decreases the conduction time from the atria to the ventricles, and due to this, more number of impulses are generated from SA node per minute, and when this occurs, this increases the rate of heart, it increases the rate of sinus nodal discharge. Number 2, it increases the conduction and excitability through different portions of the conduction system. And finally, when the impulse reaches the ventricles, it increases the force of contraction. The stimulation of the parasympathetic nerves results in release of acetylcholine at the SA nodal nerve endings. Acetylcholine, after release, it binds with the muscarinic receptors. It binds with the muscarinic receptors. The type of muscarinic receptors with which acetylcholine binds on the SA nodal membrane are the M2 type muscarinic receptors. This binding of acetylcholine with muscarinic receptors leads to activation of G inhibitory protein, and this G inhibitory protein in turn produces two effects. It produces two effects. Number one, it blocks cyclic AMP production. Number two, it activates potassium channels, and the potassium channel activation in turn leads to hyperpolarization. Hyperpolarization in turn makes SA node less excitable, and when SA nodal membrane it becomes less excitable, the stimulus needs much longer time, or the impulse requires much longer time to reach threshold potential for excitation. And due to this, two effects occurs. Number one, this less excitability of SA node leads to number one, slow rhythmicity of the nodal fibers, and number two, it slows transmission of impulses to the ventricles. The clinical significance of studying this SA nodal potential, action potential, and its mechanism is that as we have discussed, the SA node it acts as pacemaker of the heart, as the SA node generates impulses that travels through different components of the conduction system and finally leads to contraction of the heart. That is the basic function of the heart. When this pacemaker of the heart it becomes defective, it may produce one of the four effects. Number one, the heart becomes too slow. When heart, the impulse generation, it becomes too slow, that decreases heart rate. Number two, the defect in the SA node may exhibit pauses or combination of effect combination of effect 1 and 2 may occur. The fourth problem that may arise from defective synodal activity is the rare effect, that is the heart rate may become faster than normal. Thank you very much, dear students, for staying with us. Stay safe. In the next lecture, we will study about the internodal pathways AV node bundle of his. Thank you very much.